Can blood vessels and nerves survive for millions of years? It's a question that completely defies our basic intuition about death, decay, and the massive passage of deep time. When we think of fossils, we almost exclusively picture hard, heavy rocks, right? Mineralized bones sitting in museum displays, completely devoid of their original biology. But what if I told you that under the exact right conditions, the most fragile microscopic tissues of an ancient animal could literally cheat time itself?
Well, the answer is a resounding yes. The surprising reality is that delicate biological structures, like cells, nerves, and blood vessels, can indeed survive for millions of years. And this incredible fact absolutely shatters the long-held misconception that the fossil record is exclusively made up of hard, stony parts. We are finding actual remnants of the living, breathing biology of the deep past.
Welcome to This Explainer. Today we're taking a really fascinating journey into the chemical miracles that preserve ancient biology. We're going to look closely at how the scientific community is rethinking the boundaries of fossilization and how specific environments transform delicate biological matter into permanent geological records.
Okay, here's a quick roadmap of exactly how we're going to unpack this today. 1. More than just bones. 2. The rules of taphonomy. 3. The soft tissue secret. 4. Paleontology's gold mines. And finally, 5. The chemistry of preservation.
Let's dive right into Section 1, more than just bones.
Over the last 3 decades, there's been an absolute outpouring of research, and scientists are actively documenting the survival of highly delicate structures that seemingly have no business surviving deep time. We're talking about osteocytes, which are actual bone cells, and chondrocytes, or cartilage cells. But it goes even further. Researchers are finding intact blood vessels, incredibly delicate nerve fibers, and entire sheets of collagen preserved right inside fossil bone matrix. Finding these tissues intact against all odds requires us to completely rethink the timeline of biological decay.
Moving on to Section 2, the rules of taphonomy.
To really appreciate how miraculous soft tissue preservation is, we need to understand the immense hurdles an organism faces after death. This brings us to taphonomy. Taphonomy is the study of the entire perilous journey an organism takes from the exact moment of its death until a paleontologist eventually digs it up. The specific events that happen during this tiny window of time dictate what parts of an organism, if any at all, will actually make it into the fossil record.
There is a massive durability gap between different types of biological material. On one hand, you've got hard parts like bones, teeth, shells, and wood. These are highly resistant to weathering, decay, and scavenging. They can withstand being transported by river currents or pounded in a surf zone. On the other hand, we have soft parts, skin, organs, leaves, and nerves. Because these are so incredibly delicate, the rules of taphonomy dictate that they rarely persist. For soft tissues to survive, they require unbelievably rare, exceptional conditions, things like rapid burial in fine-grained lake sediments, or freezing or drying out in a cave. Under normal conditions, they're completely lost.
Which leads us to Section 3, the soft tissue secret.
So if the fundamental rules of taphonomy dictate that soft tissues decay rapidly, how on earth are we finding them preserved inside fossils that are millions of years old? Well, the answer lies in a fascinating chemical process called Fenton reactions. Recent studies have identified Fenton reactions as the plausible mechanism that allows soft tissues to literally cheat decay. These chemical reactions occur very early in diagenesis, which is the physical and chemical changes right at the start of fossilization. They're facilitated by oxidizing environments, often aided by external concretions forming around the bone that protect the delicate biology inside. It's really a perfect storm of chemistry and geology acting as a preservative shield.
Because this science is so groundbreaking, it's generated some friction. As researcher Philip J. Center noted in 2022, there are numerous misconceptions out there regarding the preservation of these tissues. Center points out that much of this stems from young Earth creationist opposition, as the survival of soft tissue is sometimes mistakenly used to argue that fossils simply cannot be millions of years old. However, impartially looking at the peer-reviewed data, it clearly shows that it's precisely these specific early chemical reactions that provide the geological stability required to preserve cells over vast spans of deep time.
Let's look at Section 4, Paleontology's gold mines, or Lagerstätten.
Now that we know the chemical secrets, where do we actually find these miracles out in the field? In 1970, scientist Adolf Seilicher coined a brilliant term for these sites, fossil Lagerstätten. Specifically, we're looking at Konservat Lagerstätten. These are highly unique rock bodies that preserve an unusual amount of paleontological information, both in quality and in quantity. You can think of these rare, isolated deposits as the highly protected, secure vaults of ancient history. They often capture animals with minimal decomposition of soft tissues, giving us an absolutely unprecedented window into ancient ecosystems.
But you can't just call any fossil bed a Lagerstätte. As the research expanded from less than 50 known sites a quarter century ago to nearly 700 worldwide today, scientists really needed a way to standardize what counts as exceptional. Researchers proposed an arbitrary but essential benchmark. To officially qualify as one of these elite Konservat Lagerstätten vaults, at least 5% of the fossils found within the horizon must exhibit exceptionally well-preserved material, like complete skeletons or pristine soft tissues.
Finally, Section 5, the chemistry of preservation.
With hundreds of these sites now recognized, modern paleontology is fundamentally changing how we evaluate these gold mines. Instead of just relying on a geographic site name, which can cause some confusion across different geological eras, scientists use 4 comprehensive pillars. They look at location, the specific paleobiology of the fossils, the taphonomy, meaning the exact chemistry of the fossils and the minerals replacing them, and the facies, which is the sedimentary geochemistry and the depositional environment of the host rock. It's a deeply holistic approach that allows for quantifiable comparisons of fossil sites through time and space.
At the very heart of this modern evaluation is the chemical composition of the fossils themselves. Whether it's aluminosilification, where clay minerals template the organism, kerogenization, where organic matter turns into carbon films, or pyritization, where tissues are literally replaced by fool's gold, these represent the exact first-order mineral mechanisms that give these soft tissues their incredible geological stability. When a soft organism dies, it's this precise chemical replacement that locks it into the rock record forever. Which brings us to a profound realization. We've transitioned from viewing fossils as just dusty rocks to understanding them as complex chemical time capsules containing the delicate cells, nerves, and blood vessels of deep time. As we wrap up this explainer, I want to leave you with this provocative thought. If highly delicate nerve fibers and collagen can survive for hundreds of millions of years through chemical miracles like Fenton reactions and pyritization, what other astonishing, impossibly fragile biological treasures are currently sitting in the ground right now, just waiting for us to find them?